So today we're going to focus on bulk RNA-seq data analysis. I will show you a typical analysis pipeline for bulk RNA-seq in analysis in Partek Flow. We will start to import FASTQ files and add sample metadata. We call that attribute. We're going to QC the data, do some cleaning up, and for the FASTQ files, we'll perform alignment to align the sequence, and then quantification will generate raw read count for each sample on each gene. We're going to clean up the data and normalize the data. Eventually, we'll perform a differential analysis to find the genes showing differential expression between our treatment groups. And at the very end, we're going to interpret the biology. Okay, I'm going to do the live demo now. So when the first time when you get the Partek URL, so that's the URL, and you're going to use your username and password to log in. When you first log in, the homepage will list all the projects you created or your colleague created, and they share the project with you. But if you don't have anything, you only see two buttons. And one is to transfer the file to the server, one is to create a new project. Whenever you need help, you can go to the help, and we have a how-to videos, and you can learn the software. Those are the short videos for each particular functions in the software. And some videos is embedded in the dialogue. You can invoke from there too. And also the documentation, we have a lot here. So we have like a quick start for you to upload the data, and there's some live training recording. I know some of you doing single cell data analysis. Today we only do bulk RNA-seq, so you can watch some videos of a single cell data analysis. Also the tutorials, and we have a user manual, white paper, etc. So you can find help from here. All right, the first thing when you want to analyze your project, you have to upload your data, or we call that a transfer your data to the Flow server. You're going to click on the transfer file and click on the transfer file button over here. And this is so you can browse your local computer. When you click on it, this is my own computer. And you just go to your folder to browse the file and add it to them. Or you can open your, I'm on a Windows, I have opened my file browser. You can select a folder or select multiple files and drag and drop and to put it over here. The bottom is the destination of where you're going to upload to. Whenever you have an account, you're going to have your own private folder. So only you can see this folder. You can create a subfolder in this dialog, and then to organize a different project. And once you're okay with this folder, you click OK. And when you click upload, the files will be uploaded. Okay. If you upload a lot of files, big files, please do not close this data tab, this browser tab. You can still working on other tabs to do other stuff, but do not close this tab. If you want to upload a big file overnight, make sure do not put your computer into sleep. Okay, so that's how you upload the file to where the Flow installed. And then once the file's uploaded, you're going to create a new project. So I will just call that a demo. I will first show you how to import the data. Then I'm going to use a pre-built pipeline to show you each step, because some analysis will take some time to do. Okay. So you create a new project, and this button allows you to add your data. And as you can see, there's some how-to videos. They are embedded in some pages or dialogues. Okay. So you can watch the video here. Click add data. First, you're going to choose what platform your data is. Is the bulk RNA-seq or single cell RNA-seq or some other file format. So today we're going to do a bulk RNA, and is this RNA-seq or ChIP-seq or DNA-seq? You're going to choose the option. And I have FASTQ files. I will choose this. And then you're going to browse to the location that you have you upload to your data. So I already have my folder organized. Once I choose this folder containing FASTQ files, on the right side, that's all my FASTQ files. And you can select them all and then click Finish. So that's how you import your data. When you import the data, first you saw there's rectangle showing you the progress of how to import the data. Once the data is imported, it will generate a circle. So that's your first data node. When you mouse over, it will tell you how big the data is. This is just a subsample of the data. I just want to show you the process. And then I select 4 files, and those 4 files actually corresponding to 2 samples. Partek can automatically recognize you have a pair and data. If you have more samples, you can click on Add Data and then continue add more samples. Okay, so that's how you import the data. Once the data is imported, and the next step is you probably want to add sample metadata, the attribute. And one way to do that is you can use like manage the sample attribute. So you first define, okay, in my sample, I have, treated and control group. So I will call that a treatment information. And 2 group name is treated and control, however you want to label. Every time when you see the 3 dots, you will have the option to modify or delete. So once you define this information, and you're going to go back to the data tab, and you're going to assign the values for each sample. So like this sample, I have, I forgot to click, so which one's treated, which one's controlled, etc. And once you finish that, click apply. So that's how you tell Partek which sample is what kind of metadata is. Okay, so now I'm going to switch to a pre-built project. Let me just show you this data I downloaded from Geo. You can see all the different tabs, and here I can keep track of the details in the project settings I downloaded from Geo. And also on this page, you can choose which user you want to share the project with you. So when you share your project, you don't need to transfer any files. The data set is pretty simple. There are 9 samples. So they are all pair end data, and they have 3 treatment groups, 5 AZA dose level at 0 micromolar, 5, and 10. I just showed you can manually create an attribute to assign that, but if you have a lot of samples, you don't want to do that. Instead, you probably already have a text file or Excel file. You have the sample name, and you have each column. I have the treatments, the gender, phenotype, genotype, all the different information. So if you have this information, the first thing is make sure in your file, in your text file, the sample name should match from here. Another thing is you can download this table as text file, and then, if I download from here, you can use Excel to add more information. So this is another trick to do it, to make sure the sample name will match, and you can add more information to the data. If you do that in Excel, make sure save it as TXT or CSV. When you do save, we don't take Excel format. You save as a TXT or CSV, okay? And then once you have that file, you're going to use the transfer file and to upload the data. Once you upload that, you're going to choose assign values from a file. And then if you already, upload the data to the folder, I already, oh, no, not this one. Where I put it? This one here. Yeah, the sample information, you choose that and then click on it, and the Partek will say, okay, which one you're going to have. And you can choose some attribute you want to use it, some attribute you don't want to use it, and then click import. Okay, so that will add this column. Once you add all the sample metadata, you're going to pretty much stay on the analysis tab. As we saw that you first have a first data node. So the full data set, there are 58 gigabytes of the data. When you so we're going to start from FASTQ file and perform some cleaning up and also alignment to quantification, and then all the way to biological interpretation. Okay, so that's what we're going to build. I will explain each step for you. Whenever you click on a circle, Partek will have the context-sensitive menu to pop up. And this is, we can recognize that this is a FASTQ file. So what you can only do is do some pre-alignment QC, you can filter, contaminate, or you can do some trimming, or you can do alignment. Okay, so you can choose all those options. And the first thing you want to do, definitely you want to do a pre-alignment QC. So I can click on that, and usually default, if you don't know what to do, default should be a pretty good option. If you know what you do, we always have tooltips to explain the information, and you just click that. As you can see, the same data node, you can perform the same task multiple times. We do not override anything. And this is what I previously performed, and this is right now I'm going to perform on it. They're going to have a different branches or different layers. Okay, now it's just showing you the progress. I just let it run, and then instead I will show you the pre-alignment QC. The pre-alignment QC does not generate the output nodes, because it really performs on this data. So whenever you see this report, you click on it, you can look at the task report, or you can look at the details. The task report depends on what browser you're using, depends on your habit. I like to open that in a different tab, but you can do so. Just close all the others. I just keep this one over here. So the pre-alignment QC will give you a table for each sample. This is the read depth, and this is the read length, average quality, no-call percentage, and the GC content. The bottom is the summary report of the quality score for each line is a sample. And this is a pretty old data, as you can see, as from the 5' end to the 3' end. Back that time, the quality dropping. Nowadays, if you use the Illumina data, they are pretty much flat. The technology keeps improving. You can click on each particular sample to look at some more detailed information. If you ever use a FastQC, it's pretty much the same visualization, but it's a Partek version. And look at the base composition, quality score, and standard deviation for each particular sample, and the quality score in a different visualization format. And this is the read length distribution. Okay. So, and this is the breadcrumb, and you can go where you want to go here. I can close this guy. And the task details is so much about the do that, open that in a different tab. It will tell you when you perform this task, how long it took. And if you have a collaborator, you always keep track of who did that. And all the detailed, input file, output file, where they are. You can click on all those links and all the parameters. In some tasks, they have lots of parameters. You will know all that and the command line. So everything is recorded, basically. But as a typical user, you only want to look at the report. All right, so suppose if you see the data is not really good, you want to do some trimming. Typically, if you do messenger RNA, your fragment length is pretty long than your read length. Otherwise, you wouldn't choose this particular prep kit. So you probably won't sequence into adapter. And typically, you do not need to remove the adapter. However, if you do small RNA-seq, like a microRNA, your microRNA is maybe only 22 base pair long, but your read length probably is 50, right? You definitely sequence into adapter. In that situation, you're going to do trim adapter. Also trim bases. I just show you the dialog, and you can trim bases. Suppose based on quality score, I want to say if the quality score drop below such and such, I will trim it off. Or I can manually trim off the 3' end, 5 base pair, or 5' end, 3 base pair, or whatever. You manually specify those information. So we give you the diagram, and then the configuration at the bottom will change accordingly. I think this should be pretty intuitive to do the trimming and then click finish. So if you want to do some trimming on your data. After the trimming, the data node still containing FASTQ file. And nowadays, if you do messenger RNA, again, you probably don't need to do this step anymore. And you're going to directly perform alignment. For RNA Seq data, the most popular aligner is STAR, and we give you different options. Or the other popular one is HiSet. Okay, so you're going to choose either one of them. If you do DNA, BWA or BowTie2 are the popular ones. But all the aligners, the dialog will be very much the same. So I will show you how STAR dialog will look like, and you will know how the other. You just need to choose where you want to align to. And so suppose this is a human data, and for we distribute a human and a mouse, two versions of assembly, like a human HD19, HD38, mouse MM10, MM39, and depends on which assembly you want to use, just choose it. If you if you want to align to other species, we do allow you to add custom assembly, and that is kind of too advanced option. I will talk about that if we have time at the end of the training. But you just choose, suppose a human, now everybody aligned to HD38. Okay, just choose HD38, align to the whole genome, and you can generate an unaligned node, which will take a longer time to do, and then click finish. If you know what you're doing, if you're very familiar with this particular task, always at the advanced option, you can click on the configure. That will give you all the tuning knobs. And we just wrap up this aligner into Flow, and we don't have an expert on this. So this is basically from their menu. And you can see when you mouse over on the tooltips, you see what the command line is. Okay. You can change all the parameters. And if your bioinformatician say, hey, you should always change in your particular design, you should always change, this parameter. You can change that and save as a new option set. So later on, you always choose that one. But most of the time, default should be a pretty good option. And after you've done the alignment, if you click on the task, you go to the task details, you will see, when I did that, how long it will take, and what is the parameter I use. And the command line is the, public domain command line, right? This will generate a BAM file. It's not human readable for BAM file. It's still pretty good. And so if you click on this data node, you're going to see my menu changed. I have a post-alignment QC coverage report, some post-alignment tools and quantification. So again, every data node, when you click on it, the circle, you will always have the option to download the data. Okay. So after you have the post-aligned data, you definitely want to check the QC. So I click on the post-alignment QC. There's nothing to configure. It just runs the post-alignment QA/QC. And then when you look at the report, again, each row is a sample. This is your raw sequence data, raw read sequence. And then this is the total alignment. One read, they can be aligned or they can not be aligned because the mistake. Or one read, you can have more than one alignment. So the read and alignment, those two different terms in Partek Flow. What you really care is about this column, the percentage of your alignment, to see how many reads are aligned. Usually, if you have more than 90%, that should be pretty good. If you see your alignment rate is around, 70%, maybe you need to go back to do some trimming on your FASTQ files and to see that if that will improve. If you have some contamination in the adapter, you can do the trimming that will improve the alignment rate. Right. However, if you say I already trimmed, my data based on the sequencing is clean, but I still have a low alignment rate, then that's probably your library prep. Maybe when you extract the RNA, you extract some other species. Your data got contaminated with other species. There's really not a whole lot that you can do. You just waste your time to sequence some species you don't care. Right? But whatever the aligned, you can still do the downstream analysis. Okay. And then those are the graphs showing the breakdown, basically to reflect the table. And if you click on any one of the samples, we give you the details. We can see this is a pair end data. Most of the reads, they have a 2 alignment, but we do see some reads that they have more than 2 alignments, right? Also, if you have a pair end data, we give you the insert size, the distribution. Basically, that's your fragment size. Also, for the aligned data, this is the aligner's mapping quality. The aligner will say, oh, how confident I think this read coming from this location. So that's another quality score. Whenever you see quality score, whether read base quality, mapping quality or variant quality or whatever the quality, the higher the better. Okay. And the different aligners, they have different skills on the mapping quality. But when you do a bulk RNA-seq, usually you don't filter your reads based on the quality score. You just, let it go. All right, so that's after you've done the alignment, we see the post-alignment QC. For RNA-seq data, now I know the reads are coming from, chromosome 10 base pair 100 to 200. What gene is in this location? In other words, which gene, for every gene, how many reads do I get assigned from the sequence? So that step, we call that quantification. So you click on the aligned data node, and then you go to quantification section. There are 2 algorithms here. One is the expectation maximization algorithm. It's a Partek own algorithm based on paper. We write the own algorithm. And there's another one, it's called HT-seq. It's a pretty popular public domain algorithm. Either one you can use. When you align to the genome, then when you quantify, you have to quantify to the transcriptome. So you need to know the gene location. We call that, quantify to an annotation model. This option, quantify to the reference, is if I align to the reference, and if my reference is really transcriptome, so suppose for some virus or bacteria, I don't have a genome, I only have all the scaffold or context, that's all the gene. So if you align to the reference, is for each transcript, you do not need to give transcriptome annotation anymore. You just quantify for, aligned to each genes, you just get the gene reads count. So if you deal with the human and mouse, those 2 are the options. When you click on this task, and Partek will ask you, I know you aligned to HG38. Okay, what annotation model you're going to use? Partek, we distribute ensemble and gene code annotation. And the annotation changing depends on, like NCBI, they change every day. Ensemble, they change very frequently. We update every 3 months. So when you start a new project, try to get the most recent one. But once you start the new project, you don't want to change it because your result will be changed. Okay, so you just choose annotation model. And those options just ask you, I would say just leave as the default, should be fine. We will only count the reads if the 100% overlap with the exonic region. If the reads is partially overlapped with the exon, we throw it away. So that's called 100%. You can change this parameter if you want to count the reads if they overhang the exonic region. Also, after we assign all the reads to each gene, each sample, we some genes, like an ensemble database, you might have 50,000 genes. They include all kinds of RNAs, like coding RNA or pseudogenes. So there's a lot of definition in this database. But probably, 10 or 20,000 of those features, they are going to be zero for your data, and it's no point to carry this over. So Partek, by default, we will say, if I look at every gene, if the total read count across all my 9 samples, they have less than 10, I will filter that out. If you say do not filter, I want to report all the genes if they have at least 1 read, you can change that to 1. Or if you say, I want to just give me all the genes definitions you get from ensemble, you choose 0. That will give you maybe 50 or 60,000 genes. Okay. And then click finish. So if you mouse over on the FASTQ file, we see this is a 50 gigabyte, almost 60, and the BAM file is roughly the same size. Those 2 data nodes containing the most data size. But once you did quantification, look at this data node, it's only 7 megabytes. Okay, so the data is dramatically reduced and is human readable now. Because this annotation, they have definition of a transcript and a gene. So we give you 2 data nodes. One is the transcript level. When you mouse over, this is a matrix. We have a 9 sample by 102,000. Here, the feature means the transcript. And this one is 9 sample with the 26,000 features. That's the gene. So at this point, if you click on it, if I download this guy, it's only 7 megabytes. You have the option. You want to put the features, means genes, on rows or on columns. Depends on what software you want to use. If you use Excel, you'd better put the features on rows because Excel cannot open that many genes, right? So if you download it, it's going to be a zip file. And after you unzip the file, it's a .txt text file. You can open txt file in Excel. So now that's how this data look like. It's a matrix. Each row is a gene, and this is from ensemble annotation. We have a gene name, gene biotype, etc. And then from column L to column T, this is the raw read count for each gene in each sample. It's a matrix. We have a 26,000 rows right here. Okay. The transcript, you're going to have just more rows there. And you probably will have questions why in your transcript, you said it's raw count, why you have a decimal point. Because if you use a Partek EM algorithm, we take all the reads into consideration. And sometimes a read will map to multiple locations. And sometimes a read map to a location, you have a multiple genes. So one is on the forward, one is on the reverse strand, right? So we split the reads using the EM algorithm. So maybe 70% of the reads belong to gene A, 30% belong to gene B. So that's our algorithm. We will have a decimal point. If you use HD-seq, you will only have a whole number. Okay. All right. And also, if you click on this circle, you can look at the task report. And the task report can also be opened by double click on the circle, if you will. That will open in the current view. And this will give you the gene level. For every sample, we tell you the total aligned reads and what is the percentage of the reads that really fully map to the exonic region based on your definition of the gene from the ensemble database, right? And this is a partially overlap with the exonic region, etc. If you do total RNA seq, you probably will have, I would say, 60 to 70%. But if you did a poly A, if you use the ensemble database, you're going to have a higher number, probably 80%. And then the bottom is the distribution of this matrix. So here's the summary of the matrix, the rows by columns. And for every sample, we tell you the minimum gene expression row count. You will always have a 0. Okay, so that's the sequencing data. The minimum is 0. We'll also give you the next minimum. It's pretty close to 0. And then you can see some genes, they got 1 million reads. So it's pretty high. Medium, 25th percentile, 75th percentile. And this is kind of a bar graph showing you each bar is a sample, and we manually bin that to say, okay, how many genes, what is the percent of the genes in this sample, they have a reads less than 10, and they have a reads between 10 to 100, etc. And box whisker plot. And the histogram, if you don't log transform it, you probably don't see a whole lot of details. Okay, so this is a gene level, and this is a transcript level page. It's a similar format, but you just have more transcript here. Okay. You know, at this stage, if you look at this table, you will notice that there are genes. So even we filter out the total count. If the sum is less than 10, they already filtered it out. But some genes, you know, the total is greater than 10. But if you look at each sample, they're very low, right? So low expression genes in any platform, in microarray, you cannot detect a low expression, differentiate that from noise. When you see a gene in this sample, there's 1 read. Do you think this gene only expressed 1 read? Or do you think that's a mistake? It's a noise, right? And also the low expression will cause a false positive. When you compare this sample versus this sample, this is a 5-fold change. Do you think that's real? Probably not, right? But when you look at the high expression genes, for example, if you have like this one, you know, I have this is 160 versus 300. This is a 2-fold change. So that's probably real, right? So I would highly recommend you to try to eliminate the low expression genes for most of the study. So that way you will avoid some false positive. So the next step, you don't have to do, but it's a good practice to filter out a low expression gene. On this matrix, you choose a filter, and the filter feature means filter gene. You can also filter sample if you think there's an outlier, right? If you know the sample is not good, when you look at the post-alignment QC, you know, I don't have many reads for the sample, you can filter out of the sample. But to filter out genes, the first choice is noise reduction, basically to filter out a low expression. But the million-dollar question is, how low is the low expression? You can see if the gene across all my 9 samples, the maximum value is less than such and such, I will call that low expression. Or if you say if the median is less than such and such, or if the total reads of this gene is less than such and such. Or another option is if you say if the gene expression is less than some number in what percent of my samples, if you have a lot of samples, when you do single cell, you're going to use this option very often, right? So you can choose your options. But for bulk RNA seq, this is what we recommend. Use maximum to filter. Depends on, you know, I will always look at this table, this table, to look at, okay, where my median or my Q1 or Q3, try to figure out a number. So half of my genes is less than or greater than 40. Maybe I will start from here. And remember, data analysis is iterative. There's no way that you can start from beginning and then you got to the end and that's it. You should always look at the result and then come back and tweak the parameter. So if you know, you pick a number, suppose I can use you know, 30. Suppose I use 30 as my criteria. What that means is if I look at the gene, look at the maximum value, like this gene will be removed because this gene, the maximum is 5, right? And this gene will be removed to 2 because the maximum is 10, right? So that will remove those type of genes. And when I first do that, I did less than 50 to filter that. Another thing to check is after you perform this analysis, look at the mouse over. How many genes do I still have? So before I have 26,000. And now after this, I got 14, 5, 6, 7 genes left. And for most of the human data, if you focus on high expression genes, I think that's enough. If you have more than 10,000, I think that should be okay. However, if you study some kind of disease, they are very, they are driven by low expression gene, you have to be careful on that. So it all depends on your study. If after this filter, I got, only 3,000 genes, I think that's filtered too much. You might want to redo it. Okay. So after the filter also will help you to improve. After you do differential analysis, you always use multiple test correction. That is related to the number of genes in the analysis. If you have more genes, you probably will have a hard time to pass FDR. Also, some analysis like Illumina or DE-seq, they are very sensitive to low expression genes because they borrow genes from other genes. So I would highly recommend you to filter to low expression, especially when you use the DE-seq, which is the most popular differential analysis method in this field. Okay, after you filter that, you can also download that. And also the data size is smaller. Before we have a 7 megabyte, now it's only 4 megabytes, right? The next step you have to do is normalization. So you choose normalization and scaling. If you don't know what to start, you can use recommended. And currently we recommend you use DE-seq for the downstream analysis. So the normalization should go hand in hand with your differential analysis, because a certain differential analysis, they assume certain distribution of your data, right? So that's what we recommended. But if you say, I know what I'm doing, I can build my own equation. So when you have a zero in your data, a lot of downstream analysis, because the differential analysis usually they are calculating the log space, you cannot log zero. So remember, you need to deal with the zero. You can add offset. I don't want to add 1, I can add a 0.1 here. And then I can do, we have FPKM, CPM, TMM, TPM, and, I add TMM. And then after that, I want to do a log transformation. I don't want to use log 2. I can use log e. And the order matters. So you can build your own equation for your normalization. It's very flexible here. And depends on the transformation. Some equations, it doesn't matter. You perform on samples or features. Like if you do log, it doesn't matter. But if you do TMM, that will make a difference if you perform on the samples or features. Okay. So make sure you know what you're doing here and click finish. So this data node is going to be the similar format, but the value will be changed to the normalized reads. And you can see the distribution of your data will be different. And this is a before normalization, the box whisker, and this is after normalization. And we can see they're pretty much, at least the median, they kind of line up. All right, so after normalization, the next step is to detect the differential analysis. It's under statistics and differential analysis. We have different options here, and the most popular one for bulk RNA-seq is DE-seq. Illumina, we have a Illumina trend, Illumina Voom. Depends on your normalization. You might not have a Illumina Voom to be available because, again, differential analysis, we kind of rely on what normalization you're using. Or ANOVA is, if you have a lot of samples, using ANOVA is a very traditional way to detect differential expression. No matter what we also have a non-parametric analysis. This will compare the rank. No matter which method you choose, the next step is very much the same. you're going to choose how you build the model. I only have one factor, so I'm going to analyze, on the dose level. You add a factor. If you have, treatment time, phenotype, genotype, this will be your sample metadata displayed here. The blue color means the categorical information. If you have numeric information, suppose I have, age, I coded as numeric, they're going to be in green color. So you choose, that's how you build a model, so only one factor analysis. And now I'm going to set up the comparisons to answer my questions. Typically, each comparison will answer, your each questions. I want to look at which genes are showing difference between 5 versus 0. Or I want to look at also 10 versus 0. Once your model is set, how many comparisons you add on, it won't affect each other. You can add multiple, or you can only do one at a time, or 5 versus 10. You can add all the combinations here. When you add the comparisons, we always want to look at the p-value and the fold change. And fold change is kind of a ratio converted. Make sure the top is the numerator, the bottom is the denominator, which means when you see the fold change positive, means that the top group is upregulated compared to the bottom. And a negative means that the top is downregulated. And depends how you phrase that. It's just the sign difference. If you did not perform a filter feature, and by default, we try to catch, some low expression genes. By default, this probably is checked. We try to filter out some genes, but you can choose to uncheck that. If you already perform a filter task, by default, we don't perform a filter on the genes again. And the advanced option will have all the tuning ups for DSeq. By default, we also give the FDR, the Benjamin and Hatchberg's multiple test correction there, but you can choose the story queue, buffer, etc. Okay, so I just use the default and click finish. And this is the DSeq result. If you open this task report, and this report is a table on the top over here, we'll see our 14,000 genes in total. And this table, each row is a gene. And then for every comparison, you will have a block here. This is 5 versus 0. We have p-value, FDR, corrected p-value, ratio, fold change. And fold change, if we don't see anything, that means it's a linear fold change. Even your data, you did a log transformation, because we keep track of that, we'll always give you linear scale fold change. You can see they're pretty big. That fold change or ratio, they are calculated from those two means, group 1 divided by the group 2. If you click on the optional column, you can add more information about, the chromosome location of my gene, or the gene biotype, whether it's a protein coding gene or a link RNA or whatever. That's all retrieved from ensemble annotation. So the next block is 10 versus 0, etc. And if you scroll down to the bottom, you can download this table as text file. Okay. All right. So for each comparison, and whenever you see this, you can choose the sort ascending or descending everything. By default, these genes are sorted based on the first p-value, which means it's this gene. If you say, I want to look at, all the genes, how the difference between those two groups. Volcano plot is a nice visualization to look at the p-value and the fold change. So for every comparison, you will have this volcano icon. By clicking on it, that will invoke a 2D scatter plot in what we call the data viewer tab. So most of the visualization in Partek will be in this data viewer. If you go to the pipeline, it's under here, the data viewer. Okay, so now you can open that directly from a certain task. In the data viewer, on the left side is the control panel. You can save this session. It's not a save a graph. You save a session, which means later on, you can you can come back over here. You can see I saved several sessions on the data. And also the setup, you can manually set up a plot. Basically, you choose what type of plot I want to make. And also from which data node, different data node containing different information. And you can see I want to use a row count or normalized count. I can make that, right? And then the bottom is some configuration, how I want to label, change the font size of my axes, and how I want to use the style to color, etc., to make the plot pretty. Okay. And here, each dot is a gene. The x-axis is how big the difference is, the fold change, but it is in the log scale. And the y-axis is also in the log 10 scale of the p-value. So basically, on the upper left corner or upper right corner of the genes, they have the small p-value and big fold change. And you can use the style to change the gene size, make it big or small. Whenever you can, on the left side, those are the modes. You have a different way to select the point. You can draw a bounding box, or you can draw a circle, or you can draw a lasso. Okay. Sorry, let me just deselect that, and I can use a lasso to draw a certain shape. So this is applied to all the other visualization here. Click anywhere else using the selection mode will deselect things. Also, when you label the genes, you probably notice that they are using ensemble ID, and you can choose, okay, I want to use the gene name or some other annotation of this gene. When you choose export the image, and that will allow you to save as a PNG, PDF, or SVG format. So this is one type of visualization for each individual gene. There's one thing I forgot to talk about that. After we do normalization, if you want to visualize the distribution of the data, another visualization people typically do for bulk RNA seq is PCA scatter plots. So you just run PCA. We call that exploratory analysis. You just explore your data, use the default, click finish, and that will generate a PCA data node. So this data node containing all the 9 samples with all the PC scores. And you can open the PCA by double click on that. That again will open the scatter plot of PCA in the data viewer. And here, each dot is a sample. And you can go to, the style. By default, we're already color-coded based on your first categorical information. You can change if you have other information. You can color-code with that too. That will help you to have a bigger picture of your data. And I can see this is my control samples. And this is a 10 micromolar. This is a 5 micromolar. So you probably will see more genes showing difference between 5 versus 0 than 10 versus 0. That's from this big picture. You can save it. As you can see, once you save the data session, they will be displayed over here. If you share your project with your colleague, you can tell them, okay, can you take a look at my, PC1, this data viewer, and that will show it right as when you save it. I made it automatically spin. You can use this control to make that automatically spin it. So like this to save. And when you rotate this, you can also capture video, and this video you can put it in your PowerPoint. All right, let's go back to the differential analysis again. I'm going to leave here. Another thing is if I want to see why this gene is on the top of my list, when I sort based on p-value, to look at in the highest resolution of the details of a particular gene, I think a dot plot is good to help you to tell the story. So click on the dot plot button. And this, you can see each particular sample, x-axis is the your treatment group. And y-axis is the normalized count. You can change this group, like I want to change how I want to do it. Also, if you change your mind, we have a lot of undo. If you go back to the undo, okay, we can do multiple undo. And this is one gene. And if you say, I want to look at another gene, one way is you can double click on this, the y-axis and search for the gene that you want to look at. So suppose you can manually type in. So suppose I just manually type in some gene and Partek will help you to search. So if I want to look at this gene, that's this particular gene look like, right? Also, if you say how I can look at it side by side, multiple genes at the same time, there's on the upper right corner, there is a duplicate plot button. If you click on that, you can drag it, say, okay, I want to make another plot, put it on the right of this plot. And for this one, I want to choose a different gene. And you can search for another gene, right? And then I can make another plot. So you can have multiple genes showing at the same time. When you export the image from here, that will export all those 4 genes together. Also, if you say, I only want to focus on this one gene, you can click on the maximize plot. And that, zoom in by scroll your mouse wheel and press your right mouse to look at the different location and zoom out. And this button is the reset button. Okay. You can select multiple plots by using control. I'm on Windows, so if you're on Mac, you can use a command, press command to select the multiple plot. And if you want to change the whole style, make all the dots smaller, or, you can change them all at once. Another thing is, as all the visualization is interactive, you can select something over here, the other plot will be selected too. All right, so that's when you visualize your particular genes, you want to tell your story, say, hey, this gene is really showing difference such and such. But if you say, I want to look at, more than 50 genes, probably this is not very efficient. And for example, I want to look at the significant genes between my comparisons. I want to present a picture that will help me tell the story. I think a heat map is a better way to efficiently help you to tell the story. So usually you want to generate significant genes by using the filter here. So you can filter based on list of gene names. So suppose you have a certain pathway, you can type in the gene name to do the filter or copy paste over here. But statistically, people like to use a p-value or FDR p-value to perform a filter. And if you see all contrasts, suppose you use FDR to specify the comparison, the all contrast means the genes have to meet the criteria for all the comparisons to meet the cutoff if you use 0.05. But if you say, I want to look at each particular comparison, I only want to look at a 5 versus 0, you will click on the per comparison. And you only change 5 versus 0. So suppose I want to use 0.05. And then here, the fold change. You use a per comparison, and you want to use a two-fold change. So that's the typical cutoff. But you should always look at your results. Now, the difference is really big. If you study, tumor versus normal, you're going to have a lot of genes difference. You probably want to tweak the parameter, use a 0.001 and fold change as maybe 5-fold change, and depends on what you want for the downstream. And we always give you the feedback, how many genes you're going to get. So once you perform this filter, you can download the table. This is a filter table, or you click on the generate filter the data node. That will generate another filter the data nodes right here. So I did several times. I generate using different criteria. So suppose I generate a 5 versus 0, and then the next time I want to look at the 10 versus 0, what you can do is uncheck 5 versus 0 and specify 10, 0.01. You don't have to uncheck this one because p-value is 1. It won't filter anything, right? Or you can uncheck that. So just to specify the criteria, generate a data nodes. So that will build the pipeline. I generate 10 versus 0, 5 versus 0, and also meet all the all comparisons. I call that a common. Every time when you right-click on the data node, you can rename that, so you will know what you're doing. The default might not be very informative to you, especially if you run it multiple times. So always change that. You will see what you did. Once you have a multiple gene list, the Venn diagram on the top upper right corner, you can click on it and just select which gene list you want to do the comparison. You can choose the identifier. Ensemble ID is really unique. Or you can use a gene name, which is more meaningful, but they might not be unique. So if you choose a gene name and make sure 50, I will make that, and then you can do all of them. So once you select them, click on the select display selection, and that's all your gene list. You click on each one on the right side, that's the genes. That's the unique for 5 versus 0. And this is the common genes for all the comparisons. You can download this gene list, or you can save in Partek. We have a list management page. You can use that to do some other stuff. Or you can use this gene list to perform a filter. So if I click on this filter, if you say, I want to perform the list on this data node, which means you will, before I have 9 samples, 26,000 genes, the raw count. But if you perform the filter over here, that will generate my key. I want to use the gene name. And the filter task will be performed. It's right here. So once you finish that, that's just a filtered gene list. And now I only have the, whatever the genes here. So once we have a gene list, this is the common gene list. If I want to present my results, those genes are really showing difference between those 3 groups. Click on that and do a hierarchical clustering. And when you set up a hierarchical clustering, you want to choose how you want to order feature, means genes, how you want to order genes. If you say, I want to use the cluster analysis, so you won't have a way to control the order of the genes. They will perform cluster and you will have a dendrogram. But if you say, I don't want to perform a cluster, I want to manually give an order, you can see the feature, the dendrogram is down. You have to give a list, a certain gene order, and then specify the order. And the sample is the same thing. So you choose what order you want to use. And also when you perform hierarchical clustering, suppose this gene list is only the results I generate the significant significant genes between 5 versus 0, I would highly recommend you only include 5 and 0 groups. Sorry. Only include those two. If you include 10, your picture won't be very, very pretty. So you can try that. So we give you a filter on the fly. You can filter include, exclude using and or combination to do complicated filtering. And click finish. And this is the results, the heat map. So some configurations, if right now the vertical is the gene and the horizontal is my sample, if you don't like this format, here is the transpose. You can put the genes this way, sample this way, and depends on how many, what your screen look like. You might want to do one or the other in order to display everything correctly. Now, if you go to axes, here, right now we display the ensemble ID, and you can choose, I want to use a gene name that will be more intuitive, more meaningful. And you can change the size of your label. And for sample, probably it's not very meaningful. You can turn it off and to give you more space to display the heat map. The heat map here, the color low expression is blue, high expression is red. If you don't like this color, you can change that. And you can also change the scale, depends on what you think that will help you to tell the story. You can also add colors. Suppose I want to add the high expression, at this value, 1.15. If there's an outlier more higher, I want to use pink. So you can have, more colors to represent in your heat map. Also, the heat map, you can use a circle or a rectangle. Dendrogram, sometimes the dendrogram can take a lot of space, and you can change, I want to make the space of the dendrogram take less space, so I can highlight the heat map. Annotation, you can annotate your samples with my dose level. So now I can clearly see my samples based on those genes that really cluster based on those levels. So those genes can really differentiate the 3 groups. And this, if you transpose, that's the one way, and this is the other way. You probably say, okay, I don't see all my genes. You can zoom in by scroll your mouse wheel to zoom in. You can see all the genes. It's because, you might not have enough pixel to display all the genes. So that's why when you zoom out, especially if you do this way, you're vertically, I don't have enough space. 1 pixel probably represents multiple genes. We just pull all the neighboring genes together. When you zoom in, you're going to see more genes, right? So, but once you zoom in, you don't see the whole picture. How can I have all that? Unfortunately, for this is determined by your screen. You have to move to a bigger screen to see the whole thing. But when you download your data, you click download. When you export, you choose export all data. You can define the size, whether in pixels, in whatever, inches or centimeter. And you can choose, I want to download as PDF. Whatever the criteria the journal needs, and you just specify the size of your picture. And now this is the whole picture. And you can zoom in or zoom out. This is the PDF. You can see this is a pretty big picture. But no matter how much you zoom in, you won't have pixels. You will see all the genes crystal clear. Okay, so that is how you present your discovery using heat map. I think a dot plot is good to look at each gene individually. That's the highest resolution to display the gene, but heat map is good to look at multiple genes together. So now we got the genes, find the genes showing differential expression between different treatment. Another thing is, so suppose when I look at my DE-seq results, those are 14,000 genes. If I know a particular gene, I'm interested in a particular gene, you type in that gene, but you couldn't find it. So probably that genes got filtered out upstream. And you will say, hey, I probably filtered it too aggressive. I want to filter, try a different filter, but I want to make it all the downstream still the same. So what you're going to do is, so you come back to this filter and redo the analysis. By clicking this task, you choose, in the task action, I want to rerun the task with downstream task. So the rerun the task allows you to change the parameter. If you say, I want to filter, if the maximum is less than 30, Partek will automatically build the downstream for you. So only one button click, you can redo the analysis. Okay. Also, when you do the analysis for each task, you can see my task probably, is a different default. It's all called filter feature. You click on this task, you go to task details. You can say, okay, this is a max, filter max less than 30. Save it. And then your task will be changed to here. Also, you can right click on a task and to change the color. So the downstream color, I want to use, some different color applied to all the downstream. So I think this color is especially useful when you collaborate with your colleagues, and you can say, hey, I want to use this color. I use the other color. So when right off the bat, okay, this pipeline is such as who built the pipeline and what parameter you used. And when you ever get to a data node, when you mouse over on this data node, you will see, I cannot move my mouse. How we get to this data node is the circle and the rectangle will have a darker border. If you see what I'm talking about, like here, they are all have a highlighted. So you know how you get to here. But if you click on it, you can go to a data summary report that will tell you, okay, I started to import the data in March, and that's my data, how big the data is, and that's all the analysis and all the parameters, they are recorded here. You can use this to help you write your method section in your manuscript. Okay, so the last step we want to do is once I have the significant gene list, I want to see, are those genes mainly fall into a certain pathway or a certain gene set? You click on this gene list and go to biological interpretation and do an enrichment analysis. You probably don't have a CAG database that require a different license, but you can definitely use a gene set database. We distribute from gene ontology database, or you can manually get the database from other places like MSeqDB, the Broad Institute. As long as you have a GAF file or GMT file, you can add your own database. And again, we update the database every 3 months too. So you choose your gene set Go database. Most of the time, Partek can figure it out whether using gene ID or gene name to match the database. But sometimes if you have a special annotation file, we might not be able to. And if that's the case, you manually specify, okay, because in the database, they only recognize the Hugo ID, which will match with the gene name. Okay. And then click Finish. Once this is done, this is the enrichment analysis for each Go term. We have an ID of the goal and the description, the type, and the enrichment score. Actually, this is converted from p-value. The analysis is using a it's not chi-square, Fisher exact test. So they only count the gene names. So basically is this table. So the p-value of the genes and fold change doesn't matter. That's why you have to filter your data based on the p-value and fold change, get the genes of your interest. Only on filtered gene list, you can perform this analysis. And if you go to the details that will display this table, in list means in your significant list. In set means in this particular gene set. Okay. And some genes, they are in the particular gene set, but they are not different. And this is the other way. This is the genes, they are not in your gene set and not significant. And you can, download the list in the gene set or in the list or not in the list. You can sort based on this, or you can filter directly from here. But if you have more in this table, you can also download the table. In this table, if you have too many rows, you cannot visualize it. So you will have this sentence to say, oh, you have too many. You have to filter down to less than 100. Either you can filter on the fly, say, okay, I want to look at the p-value less than, 0.05. But still I have too many, or I can use a 0.01, 0.001. Maybe I don't have anything. Okay, now I filter down to less than 100. You will be able to visualize. Or another way is, I will show you another way. On this gene set, you can perform a filter task. So this will give you more flexibility. You can search for a certain, gene set. If the gene description containing certain keyword, you can filter that way. Or you can say, I want to look at the enrichment score or p-value less than such and such. So you can use the or and an and operation to build your filter equation. This one is more flexible. But either way, you will generate a filtered gene list. And once it's a filter less than 100, you can visualize in the data viewer. Again, this will open two plots. And those two plots, talk, like what I said, you can select something over here, those two, they're highlighted. This one is x-axis is the enrichment score, and we sort it based on the enrichment score, and those are the gene set. Okay, just the x-axis represents enrichment score, y-axis is each individual gene set. This one is a scatter plot. It's a similar thing. But again, you can choose x-axis. I want x-axis represent, enrichment score if you want to do it this way. And I want to use the size to color by, maybe I color by enrichment score also, or color by the genes in the set. I can size by the rich factor. So there's a lot of things you can configure the plot to help you tell your story. So this kind of biological interpretation is, the idea is I look at each gene individually to detect using the p-value and the fold change certain criteria to get a subset of the gene list. And then look at the subset of the genes, are they mainly enriched in which gene set, right? So that's the idea. But the other one is, if my question is, I don't want to filter my genes. I want to look at all the genes. Instead, look at differentially expressed genes. I want to look at the differentially expressed gene set or pathway. I want to look at a pathway as a unit. If that's the case, you should start from normalized count. So you don't analyze each gene individually. Instead, you analyze each gene set as a unit. So click on the normalized data node, go to biological interpretation, GSEA is the most popular way to do this kind of interpretation. So perform a GSEA. Again, you're going to choose the Go database. And if you choose the database and then choose next, so you're going to set up the comparison. So I want to look at a 5 versus 0, 10 versus 0. This is very much like when you compare each individual gene, but it's a different algorithm. So under the hood, we'll look up the genes to see which gene map to which gene set. And then at the gene set level, we look at the differential analysis. And this analysis of the table is still, each row is a gene set or a pathway, depends on the database you have. And you're going to have 5 versus 0, 10 versus 0. And then the enrichment score will have a sign. So positive or negative, against the same thing. When you see positive, that means your group 1 is upregulated compared to group 2. By default, we sorted based on the normalized enrichment score because we take the number of genes into consideration. And there is a special visualization for each gene set. And you probably see this in a lot of publications. This is the Broad Institute GSEA analysis report. Again, you can choose the configure x-axis to represent a certain gene set. You can search for the gene set to visualize it.